This conference will now be recorded.
I wonder if Giuseppe is still going.
No, I'm waiting.
Let's see.
I'm almost done with the presentation, actually.
Yeah. What can
I lost it again. Can Giuseppe, have we lost you again? Can you test your mic?
Do you hear me now?
Yes, that's good.
Okay. Well, I'll just, I guess I'll just, I need to go back one. Okay. Okay, so can everybody hear me?
Yes.
Yes.
Okay. Okay, I'll give it another shot. Sorry about that. I apologize. This was, again, mostly intended for OLAP's information on the 5 main research activities that we're conducting at IPHC. Those 5 research activities are migration, reproduction, growth, discard mortality rates and discard survival, and genetics and genomics. The first research area, migration, has as its main objective to improve our understanding of migration throughout all life stages, from larval migrations to reproductive migrations. And the management implications are in stock distribution and regional management. The second primary research area is reproduction, with 2 main objectives. The first one is to collect information on sex ratios of commercial landings, as you heard yesterday from Ian, and to improve maturity estimates. The third main research area is that of growth, and the main objective is to improve our understanding of factors that are responsible for changes in size and age, and for the development of tools that will allow us to monitor growth and physiological condition in the Pacific halibut population. The management implications here are estimates to assist in current estimates of biomass. The fourth research area is that of discard mortality rates and discard survival. The main objectives are obvious, is to improve our estimates of DMRs in the directed long line and guided recreational fisheries. Management implications are obviously regarding the current ability to estimate discard mortality in different fisheries. And the last research area is that of genetics and genomics that it's linked also obviously with of migration. And the main objective is to improve our understanding of the genetic structure of the population and to create genomic tools such as the genome with which to conduct studies linking genetics and movement and distribution of Pacific halibut. The management implications here are in stock distribution and local adaptation. Also for, mostly for OLAP's information, we've tried over the last few years to really ensure that all the research activities that are conducted by the Biological and Ecosystems Science branch are well linked with the needs of stock assessment and the management strategy evaluation process. And in this slide, what you could see on the table below is that on the left-hand side, on the left column, you see the 5 research areas that I've described. On the column on the right is that of research outcomes, and here I've listed a few of the research outcomes for each of the main research areas, and how those, if you move to the right, how those are actually relevant for stock assessment, and which ones represent specific inputs for both stock assessment and the MSE process. So The integration between biological research, stock assessment, and management strategy evaluation is really at the core of our program. Let me move on to the actual progress in the various research activities, and I'm going to start with migration and distribution. And here I highlight the research priorities that are important both for stock assessment and as well as for the MSC process. And as you can see on the first one, in terms of the assessment biological inputs, the product really is to estimate connectivity with Western Pacific waters in order to provide information on seasonal movement across conventional lines. And the second one that is more related to MSC and below is estimates of movement between biological regions. So you'll see that these studies are actually intended to provide information regarding this research priorities. So we have actually two projects in the area of migration and distribution. The first one is on larval and early juvenile dispersal that Lori will speak to, and then the second one is late juvenile and adult migration, which Team Lore will speak to. So let's start with Lori. Lori, are you able to connect?
Yeah, I'm here.
Okay, so go ahead, please.
Okay, yeah, good morning, everyone. I just wanted to give you an update on a project that we've been working on for about 4 years now, and it has culminated in a manuscript, which we have submitted and is currently in revision. So we're pretty excited about that. Next slide. So I'll just give you some highlights here. We have our key findings. One of the things that probably our primary goal actually, that we really wanted to look at was the connectivity between basins, so between the Gulf of Alaska and the Bering Sea. And you know, it's been hypothesized for a long time, so it's not a new idea. It's just that we didn't have the tools, the modeling tools and things like that to actually sort of take a closer look at it until recently. And so now with the use of with ROMs put on top of a individual-based biophysical model, we were able to look at larval dispersal, which with some limitations, which I'll talk about a little bit, but for the most part, it was pretty eye-opening. So in the upper right-hand corner of this slide, you can see that's the actual empirical data. So this is from all the larval surveys that they've conducted since, I believe, 1982, and they don't have a standard grid or anything like that. What they do is they go out and they target different areas and so forth each year, but over time you get kind of this picture of where the larvae are actually located during these surveys. So I guess the other map that you'll need for each one of these slides, there's only a few slides, so I won't take up too much of your time, but the spawning regions one is showing how we divided up the major spawn regions as defined by Gilbert St. Pierre back in the 1980s. And so we have spawn region 1, for example, is in the Bering Sea, and then 2 is in the Western Gulf, and 3 and 4 and 5, so forth around the horn there. So the table shows that the spawn region 1, 2, 3, 4, and 5. So from each one of those spawn regions, that's showing for the 6 years that were modeled, that's showing the number of, or the percentage of larvae that made it from the Gulf of Alaska into the Bering Sea, or in the case of spawn region 1, they were in the Bering Sea already and 100% of them stayed there. So what you can see is that in spawn region 2, for example, that one in the Western Gulf of Alaska, about over the course of time, ended up or had the potential to end up in the Bering Sea. And then the further away you got from those Aleutian Island passes, you know, of course, the lower the percentage, until spawn region 5, which is over in the Eastern Gulf, just a trace amount had the potential. And so what you can see is there's 2 panels. There were a lot of these panels generated, but just to give you an example of what these panels look like, down at the bottom, so I gave you 2 years there, 2005 and 2009, and spawn region 2, so it's the one in the Western Gulf. And the colors, the blue color is where the larvae would be in 1 month. The green color is where they would be in 3 months, and the pink color is where they would be in 6 months. And these were the ones that would have been spawned in the model in January. And so you can see that there is some interannual variability, but that you get quite a lot of connectivity there. Next slide. So this is a different way of, or kind of another way we looked at it, was we wanted to know, once they get into the Bering Sea, where were they going? And so this was an attempt to look at that. We partitioned out the Bering Sea into domain regions, including the Chukchi Sea, actually, which is A there at the top, and the Gulf was its own region. And so we were trying to see if we could get them inshore, or how many were going across the pond to the Russian coast, that sort of thing. So this, if you were here last year, you might remember this, this particular graph, and you could see that each one of the squares up above, the 1, 2, 3, 4, and 5 correspond to the spawning regions, and then along the x-axis are the years. And so one of the things we wanted to look at in the Bering Sea, they have warm stanzas and cold stanzas, or have been having those in the past 15, 20 years. And so we looked at 3 years in each, wanting to see if there were distinctive differences within those two stanzas. And what we found is, while there definitely are interannual differences, we didn't really see anything jump out at us between warm and cold. Next slide, please. And so another thing we wanted to look at, which I thought was really interesting, large degree of intra-basin connectivity. So within basins, so the biggest thing is we know, or we think we know. So these were all things we thought we knew. So in the Gulf of Alaska, not a surprise, the panel at the bottom is the spawn region 5. So that's the one on the eastern part of the range there. And those stay in the Gulf of Alaska, but basically spread across the Gulf of Alaska, and also southward into our area 2B and maybe even 2A. And then spawn region 1, which is the one in the Bering Sea, you can see, and I'm giving you 2 sample years here, 2005 and 2009. So you can see the difference in years, but in both years, what you're seeing is a lot of connectivity between the eastern and the western Bering Sea. Next slide, please. And then Ray Webster may be able to speak to this better than I, but to give in a nutshell, the other thing we wanted to look at was, what are these fish doing after they have been advected onto these nursery grounds? And we have done a lot of tagging and so forth, being the IPHC, but the young ones are pretty elusive because they're hard to tag, they're hard to find. So we looked at ages 2 through 6, and on the right-hand side, you can see there's a sample panel there. This is the 2005 year class, so at age 2, age 4, and age 6. And the lighter colors are where the fish are found. And so you can see that they begin in Bristol Bay, and they move out. In 2005, they moved out pretty much as a kind of a clump, but then by age 6 are pretty well dispersed. And then the same goes for around Kodiak Island. You can see them when they're very young, right near shore, and then they kind of disperse outward from there. Next slide, please. And so the kind of the big conclusion was that, yes, we could definitely see this connectivity between basins, and in fact, a circular life stage connectivity as well. So the advection into the Bering Sea, and then we did see with something I didn't point out in the last slide, but we did see that the fish do move pretty steadily down towards the passes, and we do have tagging data from some of our fish that are a little bit older, 5, 6, 7, that we do show them actively moving out of the Bering Sea back into the Gulf of Alaska. And I think that is the end of that.
Thank you, Laurie. Continuing the studies that Laurie described, as you know, we've been tagging slightly older fish than the one that Laurie was describing. We're tagging fish in our setline survey, so we're tagging fish that are at least 5 to 6 years of age. And an update on the numbers of tagged fish since 2015, when we initiated this project. we've tagged 11,703 fish that are under 32 inches in length, both in our own survey as well as in the NIMS trawl survey, of which we've recovered to date 146 fish. And in the map on the right, you see with the blue triangles the site of recovery of those fish that were released. And you know, if you look at the Gulf of Alaska, the detail, I'm sorry, is not good enough for you to see, but most of the direction is from a western to an eastern location, and from a northern to a southern location, which is consistent with the patterns of migratory behavior in fish that are of that age. So now I'll give the word to Tim Lohr, who's going to describe studies on adult acidic halibut. Go ahead, Tim.
Good morning, everyone. Hopefully going to be an update on our northern Barrier project.
Tim? Bad audio.
That's bad audio.
Is there a way to improve?
Tim, can you actually start again, speaking a little louder?
Yeah, I can. Can you get me now? Is this good?
Yes.
A little better, but there's some static. Go ahead.
Yeah, I'm not sure what I could do about static. I'll just have to bear with that. I'm just going with the microphone. Okay, actually, I got to move to a different room. One moment.
Tim, we cannot hear you.
Okay, is this audio better?
Yes.
Yes.
Okay, change machines. Okay, so this is mostly going to be an update on our northern Bering Sea tagging that we've been doing for a couple of years. I want to give you a little bit of background on our broader Bering Sea research that started in essentially 2008, because I don't believe I've given you guys any of this, and I certainly haven't given it to Olaf. So we have a fair history of satellite tagging, pop-up archival tagging in the Bering Sea that again began in 2008 and also 2009. We also had some releases up on sort of the upper side of that near Neverin Canyon on the US-Russian border in 2016. And the data extend essentially to last summer because we did have tag recovery in 2019 as well. So over that period of time, we have data from 145 fish. We tagged more than that, but 145 fish for summer to summer pop-up tagging, where we tagged the fish on survey at our survey stations along the Aleutians and up the shelf edge. And those tags were recovered and reported their data essentially a year later. So that's the distribution that you're seeing here. Next slide. So we get a few different things from this. These are the endpoint location data. So the solid dots being where the fish were tagged and the open triangles where the tags reported a year later. We get evidence of sort of regional differences or regional substructure in movement patterns. Fish in the Aleutians in particular, both in the western and central Aleutians, remaining fairly resident from year to year, kind of separated by the deep passes, and Chicka down the middle and Amukta on the right, with considerably more interannual dispersal of fish that are tagged along the shelf edge, both within the Bering Sea and south of Unimak Pass in what I've got listed here is the transition zone, which is essentially southern 4A and the Aleutian Islands, the eastern Aleutian Islands. There's a fair amount of movement along the shelf edge that includes movement into Russian waters. We had fish go as far as Gulf of Anadyr, and last year that tag that was recaptured somewhere along the Kamchatka coast. We don't have a good endpoint on that, but we do know it was coastal Kamchatka. And then movement from southern 4A as far east as Washington, and a fair number of fish moving into Area 3 as well. So next slide. Along with that, we can get evidence of movement while those fish are at liberty using light data. The tags record light, and so from daily light curves, we can get estimates of longitude. Latitude doesn't really work for halibut. It does for pelagic fish where you can infer day length, but you can't really infer day length for really deep water fish. But we get some good longitude trajectories. The fish on the left is a fish that it's not a annual fish. Its tag popped up in the middle of winter, but you can see that its movement was timed in late fall, early winter, December, January, which was coincident. The lower panel is its depth distribution. So it moves into deep water before it starts moving eastward, but as it starts to move to its sort of really deep water phase, it slides across and into the Gulf. And the central fish is a fish whose tag popped up in 4A. So from the endpoint data, it doesn't really look like the fish has gone anywhere. But from the light data, you can tell that along with that depth trajectory, it moved eastward into area 3A. So you've got seasonal movement in the fall out to 3A, and then seasonal movement in the spring again back to 4A. And the far right panel is just sort of all of the data points for all of the fish that were tagged in 4A. And you can see sort of that distribution of point out to the east, which indicates both the seasonal and interannual movement of fish that were either emigratory full-time or just seasonally. And next slide. And so this sort of expands those individual depth trajectories to look at depth-specific movement for each of the sort of 5 biological or ecological little regions that the movement data indicated were sort of spatially variable. On the top are all of the fish that were tagged along the Aleutian Island chain from southern 4A out to the western Aleutians. And I would draw your eye to the notion that most of these fish are in deep water in mid-January, which is what we've been setting as sort of our midwinter pop-up date for pretty much all of our studies based on early data from the Gulf of Alaska that also demonstrate that the fish are mostly on their spawning grounds by mid-January. By contrast, in particular, the green line on the bottom, which are the fish that were tagged in shallow water on the eastern Bering Sea shelf around St. Matt and around St. Paul, although they're shifting into deep water sort of in December and January, the real deep water phase is shifted out sort of toward March, April. And when we get high resolution data, we get these tags back and we can look at the minute by minute or 30-second by 30-second data, it suggests that the active spawning when they're moving into the water column to release eggs is in that sort of deep water phase. And so it looks like the eastern Bering Sea shellfish have a shifted timing relative to the Aleutians and even the Gulf of Alaska, and that will become important in a moment as we move to the next slide, which is the work that we started in 2019. What we don't have is very much information for fish tagged on the eastern Bering Sea shelf at all, and in particular in the sort of Norton Sound area, which is the upper right-hand point of those 2 red lines. The local fishing group, Norton Sound Economic Development Corporation, noticed a lot of cod moving in starting 3 or 4 years ago, and halibut have started flowing in behind them, presumably due to increasing water temperature, sort of a climate change range expansion effect. And so initiated by them, we've begun tagging in that region. It's a collaboration between NSEDC, which was providing the majority of the funding for the logistics and the tags, University of Alaska Fairbanks. We've got a graduate student funded through a Rasmussen Fellowship who began last year, and he should be on the project through 2022 anyway. And the tagging is mostly around Nome and Savunga. Next slide. We started last year, got 44 fish tagged, sort of 3 different platforms. The red dots are fish that were tagged off the NIMS Northern Bering Sea Troll Survey, which won't exist this year and may not exist again. In Norton Sound, the green dot, we chartered a vessel to deploy tags out there in July. And then the blue dot is out at St. Lawrence Island, is one of the native fishing communities at Savunga. In August, NSEDC partnered with them to get tags out on the local fleet using their effort. The tags were reported to pop up in 3 batches, mid-January this year, summer this year, which will be July and August, those should be coming up, and summer 2021. And so next slide. So our only pop-up group so far was this January, and it was relatively disappointing in that we only got 4 out of our 14 tags. Now it's normal for some of your tags to go silent to not function, but 4 out of 14 is a very low reporting rate for these tags. They have a really high reporting rate in most environments. And what we believe is likely to have happened, we've got those 3 fish that moved out toward the shelf edge from Savunga. You will notice that they did not make it to the shelf break by mid-January. One of the smaller fish was the U32, those are large fish. The U32 moved down to around Shalikoff Strait down in Kodiak. And next slide. What appears to have happened is that the relative disappearance of most of the tags is that they were scheduled to pop up essentially under the sea ice. We unfortuitously happened to tag fish in what was one of the largest ice advance years in the Nome region in over a decade. There is some evidence, apparently 2 weeks ago, one of those mid-January tags pop up under the sea ice. They should detect that they're still in water. They should not broadcast. They should start broadcasting when they melt out. And apparently one of them did melt out, the graduate student reported about 2 weeks ago. So hopefully we will get more of the environmental data back as they pop up. Obviously the location data won't be very robust. They certainly won't tell us whether, where those fish went. Using the location and the temperature data, they may indicate sort of certainly whether or not the fish had remained in the area under the ice, but it's going to be kind of a really coarse scale analysis there. So next slide. We will be continuing this year. NSCDC now has sort of 50 tags that have been purchased that are in production and will be shipped within the next week. And there are 12 tags, sorry, that were left over from last year that did not go out. So there's 62 tags completely. We'd expected it to be a pretty ambitious deployment year. It probably will not be because of the COVID issues. At the moment, nobody can get into or out of Savunga unless you're a resident or emergency personnel. And so we don't expect to be able to get to Savunga at all this summer. Nome is on a now 1-week quarantine. You have to get a positive test upon arrival, 1-week quarantine, negative test at the end of that, and that includes arrivals from all locations. So even our graduate student from Anchorage cannot get from Anchorage to Nome without quarantine. And so we're probably just pulled the plug on that, and he probably won't go at all unless things change. So we do have the one person employed at NSCDC who has partial halibut responsibilities, and she will be attempting to deploy as many tags this summer as she can get done. We again assume that will be no tags in Savunga and probably not the full suite in Nome. So I'll be happy and impressed if she gets sort of 24 tags maybe deployed this year, which is going to leave a lot of tags left over for next year when NSCDC is also planning to buy another batch of 50 years. So 2021 will probably be our biggest deployment and give us data stretching out into 2022. And there might be another slide, or that might be it. That is it.
Thank you, Tim. The last project really that we're conducting in relation to migration distribution is one that I'm just going to provide you with the update of numbers of fish that have been photographed. This is what we call the tail pattern recognition project. The objective is to use natural markings on the tail of Pacific halibut to identify individuals over time and inform on movement patterns and growth. This is at a stage where we're basically just collecting images from fish that are under 32 inches, fish that are also wire tagged, and so far we have collected images from 936 under 32 Pacific halibut, and of which we have recovered 14 tags with tail pictures. The idea is to build a library of images both at the time of release and at the time of recovery in order to see whether those markings are stable over time. And hopefully we'll be reporting on that in subsequent years. So let me move on to the next area, which is reproduction. And here we have two projects that I highlighted in the objectives. One is the sex ratio of the commercial landings, an important input into stock assessment, and as well as the updated maturity schedule. So in order to do that, let me show you the first. I'm having a little difficulty advancing the slides. Yeah, here we go. So the sex identification in samples from commercial landings is conducted with the use of genotyping techniques based on tissue that is collected in the form of small fin clips from the tails of Pacific halibut that are collected in the ports, and then we use two different SNPs or single nucleotide polymorphisms assays that are TACMAN-based to identify males and females through genetic means. And you've heard that we have completed the entire set of aged of 2017 and 2018 commercial samples. Those were included, as Ian indicated a couple of days ago, on the 2019 final stock assessment. And we're currently working, and that's the work of Anna Simeon, our laboratory technician. She's currently working on the 2019 commercial samples. We're processing over 10,000 fish per year. So hopefully we'll have the sex ratios for the 2019 commercial samples for this year's assessment. The second project is the revision of the maturity estimates, and for that, one of the main objectives was really to understand the basic concepts of reproductive biology in the species, and for that we started the characterization of the annual reproductive cycle to improve current estimates of maturity in both male and female Pacific halibut. I will only show you the results that we have so far on female maturity. We conducted between September 2017 and August of 2018 monthly collections of biological samples. Each month we collected biological samples and information from 30 males and 30 females. With these samples we conducted obviously histological assessment. That was one of the key things in order to improve current estimates of maturity, but also we collected biological samples such as blood or tissues, pituitary and gonads, in order to conduct analysis of specific reproductive markers, as well as energy levels in the form of fat content or the hepatosomatic index, which is the liver weight relative to the overall round weight of the animal. One of the main things that we wanted to do is really to revise the scoring criteria that is currently used in our Fisher's Independent Setline Survey through the macroscopic observation of the gonads in the field, macroscopic or visual observations. So the deliverables that we are expecting from this project is an accurate staging of the reproductive status of both males and females, to update maturity at age estimates, and provide, if any, and we still are in the process of examining the possibility that there are there is skipped spawning in Pacific halibut females. One of the first objectives is really to revise the maturity estimates by comparing macroscopic versus microscopic maturity stages. Currently, we're using in the field 4 maturity stages, you see here, 1 to 4, from immature, maturing, ripe, and resting. 2, 3, and 4 would be considered mature in stock assessment. But we also want to conduct maturity staging based on ovarian histological assessment. So basically, conducting histology on the ovaries, classifying the stage, the developmental stage of the oocytes, and then assigning a particular maturity stage based on the most advanced oocyte, and then compare the two methods in order to revise, if necessary, current microscopic maturity stages. This is what we obtained in that full calendar year sampling. So here you have on the x-axis the months between January and December, and on the y-axis the proportion of mature females, and in the colors you see in white, in gray, the proportion of females that are classified as immature in the field, and then in pink as maturing, red as ripe, and 4, which is that burgundy color, it's resting or spent. So one of the interesting things to highlight is that during the months that we're conducting the survey on, which is between typically between June and late August, beginning of September, that's the time when you actually start seeing an increase in the number of females that are maturing. So look at the pink lines. By July and August, more than 80% of those fish are actually maturing, and those fish will likely, most likely, contribute to the spawning that will take place later in the season. So this is the time when really the survey takes place. That's when we collect the information on maturity, and this highlights that this is really a relevant time to be collecting that information. And again, yes.
Excuse me, could I, I'm sorry to interrupt, could I just ask a question based on this slide?
Yeah.
If you were interested in using the macroscopic maturity staging as a means of identifying skip breeding, what would you expect to see? What would be an example of a distribution where, or a stage at a specific time, month, that you would say, okay, this is indication of skip breeding of this particular female or a group of females?
Yeah, that, if you look at, yeah, that's something that we cannot really provide the information right now because we're in the process of assessing that. But if you look at December, and you see that there's about a 30% of a total of 30 females in that month, there's about 30% of those females that are labeled as immature. The remaining females are maturing in December. What I'm really interested in is determining whether those females that are classified as immature, if they are really immature, or we are actually misclassifying some of those immature females that are skipped females, based on, and this is just based on the external appearance of the gonad. The answer to your question, I think, will be when we finally look at the histology and try to determine really, you know, the accuracy of this maturity stages. But, you know, I would be very interesting to see if in December or maybe earlier, maybe some of those females that are classified as immature could actually be skipping.
So the immature stage is solely based on the gonadal stage and has nothing to do with body size?
That's correct. That's just maturity stage. We have not yet incorporated size or age into this classification. That is the next step.
Thank you.
So let me continue. And one of the things that we did in order to classify females according to their histological status is to really describe the oocyte developmental process. So we conducted histological analysis, and we classified Pacific halibut oocytes in terms of their developmental status from primary growth, and here you see 3 examples that are increasing in developmental stage, from 1 nucleolus to cortical alveoli, secondary growth, and those images of the oocytes here are not respected to size, but you will see in a minute that there's a significant increase in the size of those oocytes, and this is because of the growing and continuous incorporation of blood constituents inside the oocyte, mostly in the form of lipoproteins. And then finally, you have oocytes going through the oocyte maturation. So here you see germinal vesicle migration, germinal vesicle breakdown, and then the hydration of the oocyte, which is really the step right prior to ovulation. So if you look at this graph on the right, on the x-axis, we have oocyte diameters from 0 to 2,000 microns, so that's 2 millimeters. And then you here see in the y-axis the oocyte developmental stage, from the least developed on the bottom to the most developed in the top. And what you can see is that there is a gradual increase in the oocyte diameter that corresponds with the advancement of the oocyte developmental stage. With mature oocytes, basically pre-ovulatory oocytes, reaching sizes up to 2 millimeters or higher in diameter. When you look at the oocyte size distribution at different stages, so each of these lines represent females that have been assigned to a particular maturity stage based on histology, and that is based on the most advanced oocyte at that stage. So, for instance, females that are classified at the perinuclear stage, that means that the most advanced oocyte in that female was at that particular stage. So when you look at the size distribution of oocytes present in those females at those reproductive phases, you see that there's a motile phase. There's basically one cohort of oocytes that is beginning to grow in size in the next developmental or reproductive phase. And then starting at the vitellogenic 1 stage, then you start seeing the appearance of a second cohort that, with the advancements of the reproductive phase in those females, then start separating, increasing in size, and then fully separating from the first cohort in the late vitellogenic, what we call vitellogenic 3 female. And then those second cohort now advances in terms of diameter in the germinal vesicle migration stage. And then in the periovulatory stage, so these are females that are really pre-ovulatory, then you see, in fact, evidence for a wide distribution of large oocytes, some of which are still on that germinal vesicle migration phase, but some are clearly have clearly hydrated and are getting ready for ovulation. So what we see is that appearance of 2 cohorts, one that always remains at about the same size, and then a second one that advances with maturity. And this is typical of a group synchronous reproductive strategy. And the fact that prior to ovulation we see a wide distribution of oocyte sizes, that is evidence of some oocytes being moved towards ovulation, and that is a typical feature of batch spawners, as the Pacific halibut is. So with this oocyte classification criteria, now we've looked at the temporal changes in the presence of females at those stages from January on the left to, I'm sorry, in this slide, it starts in March and then it finishes in January and February. What you see is that in January and February, that's the only time really when you see pre-ovulatory oocytes, and that coincides with the idea that January, February is the peak of the reproductive of the spawning season, at least in that area in the Gulf of Alaska where the samples were collected. And that in August, September, and October, that's when you start seeing fully grown vitellogenic oocytes. Those are the late vitellogenic, what we called before the vitellogenic 3. And if you look at the second graph here, the one that I'm highlighting with the cursor, this is the gonodosomatic index. And if you look at when the gonodosomatic index starts increasing, that's precisely the time of August and September when you start seeing a high proportion or a growing proportion of late vitellogenic females. So clearly, there is a process that has now been clearly identified through histological means. One of the things that we want to do, in addition to continue, working on establishing the correlation between macro and microscopic staging, is to look at possible temporal changes in maturity. In that portlock area where the samples were collected initially, we collected samples there in June of 2017, again in 2018, in 2019, and this year we're also going to collect samples in in June of 2020 in order to have 4 years or 4 consecutive years of ovarian maturity estimates through histological means and see if there's any temporal changes. Probably more importantly is the fact that this year, for the first time, we're going to start looking at possible spatial differences in maturity. So now we're going to sample at the same time in the beginning of August 5 different locations throughout the Gulf of Alaska area, as far south as Hawaii and closer to Kodiak. So with this information, we'll have an idea, a preliminary idea of whether there's any spatial differences in maturity in females collected at the same time. Let me move on to the third area, and we're getting close to finishing. This is an area that is really related to trying to understand the historical changes that the Pacific halibut has experienced in terms of size and age, and whether changes in growth, somatic growth specifically, have any information to add to this process. We conducted a study that I described to you earlier, and in which we conducted captive studies on juvenile Pacific halibut and manipulating growth rates through temperature manipulations, through density manipulations, through hierarchical dominance, and capture stress. And I'll present to you today the results that we've obtained through temperature-induced manipulations, because we've been looking at potential signatures in skeletal muscle of temperature-induced growth manipulations that could be used to derive information on potential markers for somatic growth in Pacific halibut. So we've been looking at how low temperature acclimation, so basically comparing fish that are acclimated at 9 versus a low temperature of 2 degrees centigrade, how this significantly decreases the standard growth rate, or how a change of temperature from low to high in fact causes temperature-induced compensatory growth that results in an increase in the specific growth rate. If you compare the red bar at 9 degrees versus the green bar that represents the temperature-induced compensatory growth group. So what we did is to collect muscle samples from these 3 different groups of fish and look at potential differences in genes and proteins that are expressed in skeletal muscle, again, in order to identify potential markers for growth that could be applied for field studies. These are some of the results comparing the 2 growth manipulations. So these are Venn diagrams indicating the number of differentially expressed genes in response to the temperature-induced growth manipulation. So if you look at here on the left-hand side of the screen, we're showing the transcriptomic data, so gene expression, and in the blue circle we have identified 844 genes that are downregulated during growth suppression. On the other hand, we have identified 223 genes that are upregulated, which means their expression is increased when growth is stimulated during that growth compensatory growth phase. And then we focused actually on the number of genes that are shared, meaning genes that actually behave their expression pattern in parallel to the growth manipulations. And we did the same with the proteins that were expressed. These are differentially abundant proteins in skeletal muscle from the same fish, and we identified 117 different proteins that were downregulated with decreased abundancy in skeletal muscle in those fish that were growth suppressed by low temperature acclimation. And in contrast, we identified 116 proteins that were upregulated with increased abundance in those fish that underwent compensatory growth, basically growth stimulation. So what we did is to focus really on the common genes initially. So we identify 59 genes, again, that their expression pattern mimics the growth response. And a number of these genes, in fact, are involved in contractile elements as well as in protein synthesis. Very similar to what we find with the proteins. Now we're looking at commonly expressed proteins that behave their abundance parallel to the growth manipulations. And here we found 33 different proteins, mostly involved, as you see, in protein synthesis, translation, and also in the contractile machinery. So a next step of analysis was to, in fact, look at what proteins and genes actually responded the same way to the different growth manipulations. So we tried to ask the question, how many of the same genes and proteins are actually downregulated when you suppress growth? And we identified 23 different genes that are decreased in expression, both at the mRNA level and protein level. And in contrast, in red, here you see the genes and proteins that are increased when you experience growth stimulation. So those are genes that change both at the mRNA level and at the protein abundance level. So a total of 10 different genes and proteins. And we finally identified 2 that were actually present in both comparisons. So there's 2 genes that change at the mRNA level and the protein level, and that are both involved in protein synthesis. One is asparagine synthase, and the other one is ornithine carbamoyl transferase. And those are potential markers for temperature-induced growth manipulations that we're going to be validating in the subsequent set of studies. So this subsequent set of studies is now not in juveniles, but in adult Pacific halibut that we have in captivity currently in Seward, in a study that is done in collaboration with Alaska Pacific University. And we're doing dietary manipulation of growth in order to derive different growth patterns and see whether those markers that we initially identified are also present in this adult fish. So I'll pass the word to Claude Dijkstra, who's going to talk about discard mortality rates and survival assessment. Go ahead, Claude.
Could I just ask a question, Joseph, first?
Yes, yes, absolutely.
You know, the gene anthologies that you showed, I mean, it's really, really cool work to get to this point to look at genes that might be under selection. I wonder, though, you have the genome. Have you tried to find these genes in the halibut genome? I noticed that the gene anthology were from a chicken and for a human, and one has to be careful about placing too much emphasis on particular genes that are based on searches that come up with very taxonomically different species.
Yes, thank you, Kim. That is a really good point, and that annotation will probably be redone once we have a fully annotated genome. Andy will talk about this in a minute. But the answer to your question is that we have not done that yet. This is something in our mind, and we've discussed that with Andy to map some of these differentially expressed genes and proteins to the genome. But what I can say is that the mapping of all the RNA seq data to the genome has been fairly successful. I think, and Andy will correct me if I'm wrong, but I think 98% of all RNA seq contexts map onto the genome. So a really high percentage of transcripts map to the genome. But we have not done that for those particular potential marker genes, but we will do that. And
Okay.
Hopefully we can provide that information by the next SRB meeting.
Okay, thank you. Just a really quick follow-up question. Has, in the intersessional period you and Andy shared some data on loci from a SNP panel that was developed to look for the sex-linked marker, and there were a number of genes that were, or SNPs that, based on higher differentiation between populations, were potentially genes under selection. Do you have plans to see if any of those sequences of those SNPs are likewise genes which you've identified in your transcriptome analysis?
Yeah, that is a really interesting suggestion, and we will definitely do that. We have not done that yet.
Okay, thank you.
Yeah, thanks.
Giuseppe, just a quick question.
Yes, Sven.
As Kim says, it's really interesting work ecologically, but presumably these are quite transient effects, right? You might be able to notice, I don't know, what sort of time periods over which you would be able to recognize these during the change. I mean, you've kept conditions constant, but obviously out in the real world, they aren't constant. So how would you be able to get a long time series to see when growth has been suppressed and not suppressed, which ultimately is what's going to affect your weighted age? I'm just not quite sure how you intend to use the information.
Yeah, no, this is again, it's a good point, and it's highly relevant to what we're doing. Yeah, this represents a snapshot on growth-related genes at the time of capture, obviously, and at the time of capture and probably recent growth trajectories. So it basically represents it gives a good snapshot both at the gene expression level, at the protein level of the growth trajectory of that fish at that time. You know, prior growth patterns that those fish may have experienced, it's probably not going to be addressed through the use of this strategies by looking at you know, genes and proteins. There's another strategy to look at earlier events during the life history of those fish, and that's through the use of epigenetic modifications of the genome, because that has been described in the literature that early events that have consequences on growth leave specific marks on the genome, and that affects the expression of those growth-relevant genes. So the answer to your question is that these patterns are looking at the current state of the growth regulatory machinery at the time of capture. So it provides a snapshot of that growth trajectory at that time. So we know whether that fish is actually you know, on an upward growth trajectory or is it a downward growth trajectory. But prior growth history, that it cannot be determined, I think, from this kind of approaches. We would need to use something else.
I know I'm taking a lot of time here, but if I could just spitball something at the group, could you go back to slide 37 and then might address at least a hypothesis for Sven's idea?
Yeah. Sorry, I'm doing this through the mouse. 37, you said?
Yeah. So, you know, the experimental design at focus on the temperature range may not reflect reality of where these animals are at that particular time period. But okay, well, yeah, what I was looking at is maybe it was before, but you can see that your treatments were 2 degrees, 9 degrees, then 2, 2, 9, right? And then
yep.
you referred to the 2 to the 9 as compensatory growth of something. And I remember in a part of the biological report dealing with migration, there was some discussion about what, you know, newly hatched, or not newly hatched, but dispersing larvae, right? They're in a spawning area, and those are really great depths and it's really cold temperatures, and they move out of there, right? So that could be, in this context, maybe a 2-degree group. And they disperse and they warmer water, and that could be probably not 9, but something a little bit above. And so really by a larvae moving from a cold, deep area to a warmer area, maybe that's an indication of something that might develop into a what you're calling here in your experimental sense, elevated growth, right? So that might be a very positive thing for a larvae to do. But you notice that you discussed that perhaps with warming, they're less likely to move, or they're less likely to move at a lower time. And so it could very well be, again, it's a spitballing argument, that warming temperatures may disincentivize larvae from dispersing. And if they're not doing that, or maybe not doing that as often, then there might not be this elevated growth that would be realized from moving from a cold place to a warm place. So just an idea.
Yeah. Yeah, thank you, Kim. That is a good point to take into consideration.
But Kim, would you not have expected that? I mean, there are energy demand elements in this as well. You aren't going to get that compensatory growth unless the additional food is available. And my feeling would be, from an ecosystem impacts perspective, the changes in productivity associated with those sorts of temperature changes are going to, well, global warming temperature changes are going to be far more dramatic than the metabolic changes within an individual that you're going to see. So I still don't quite understand how this is going to help the stock assessment, but
well, I mean, what it might suggest is if there's declining size at age, it could be occurring at a very formative early ontogenetic stage, and it could be occurring because of the metabolic differences between and foraging, right? It's warmer, you're metabolically more active, you're feeding more, and I don't know. It's just something that might be worth thinking about.
Yeah, thank you. If there are no more questions, maybe let me move back to where we were, and okay. So yeah, I'll let Claude speak to this next section. Go ahead, Claude.
Good afternoon, everybody. Hopefully you can hear me. So I'm going to speak to the work that the IPHC is currently doing with respect to looking again at discard mortality rates and survival assessments. And we've got 2 kind of prongs on this right now. One is focused on improving DMR estimates in the directed longline fishery. So these are U32 fish that don't fit the size regulations, so they're released by the fishery. And the second one is starting some investigations into DMRs in the guided recreational fishery. Next slide, please. Are you able to hear me?
Yes. Yep.
Okay, thanks. So in late 2017, we did a field project where we went out and fished using stuck gear, so the same gear that's used by the FIS program. And we looked at the handling practices and injury levels and physiological conditions of the halibut that were captured and released. The treatments were careful shake, ganglion cutting, and then hook stripping. Hook stripping is not it's not allowed in the regulations, but occasionally fish make it past the person at the roller, and the fish gets hit by the hook stripper or crucifier, as it's colloquially known in the fishery. We collected injury information on those fish. We took blood draws. We did some fat readings, length, weight, all that sort of stuff, as well as recorded environmental conditions, things like time on deck, temperature differences between them and the water up on the surface, and all of those things. Some of those have been previously reported on, and I'm kind of focusing on things that have been developed since our last SRB meeting, which is the primary item for that is we've worked up all the physiological reads in the lab, and we're starting to analyze those now. So next slide, please. So just briefly on the injuries, the first panel there on the left shows the injury proportions by release methods, and the injuries are listed off to the right there. And so you can see that ganglion cutting and shaking have a fairly similar injury profile, with the largest proportion of injuries being simply a punctured or a torn cheek. So that's where the hook protrudes through the cheek. And then the hook stripper has a much broader suite of injuries that include a lot more kind of severe injuries that you would see on the lower end of the codes on the right there. So things like torn faces, eye injuries, a torn cheek and jaw, things like that. So we get kind of a fingerprint of injuries for each of those release methods. We also looked at what is the release condition or the viability of those fish. The viability is analogous to what is used in the observer, the observer world, where they grade the fish out as excellent, moderate, poor, or dead through a key. And so you can kind of see how these injuries fell into each of those bins. So the majority of fish that have torn cheeks typically get shunted into the excellent category, but some of them still do make it into that dead category as well. So next slide, please. We are also able to look at how each of the treatments, so the method that the animal is released from the hook, how they bin out into those release conditions, the excellent, moderate, poor, and dead again. Here again, you see a trend where the hook stripper, as would be expected, creates more significant damage and leaves more of the fish in a poor condition when they're being released. So we had over 1,049 fish that were released with a wire tag in this study, and 79 fish were released with a satellite tag on them, and that stuff's been previously reported. And then we also obtained about 570 physiological measurements from the wire tagged fish. Next slide, please. Here's some of our initial findings with those physiological measurements. So along the x-axis, you have the release condition or the viability of the animal, and on the y-axis, you have the physiological element of interest. So panel A is their glucose levels. We didn't find any difference in glucose across the release conditions. Panel B is the lactate, and we did have a significant difference on the amount of lactate in the dead animals, but there's pretty high variance on those levels in general. And then panel C is the cortisol levels in these fish. And again, there was no significant differences between any of the release conditions of those fish and their cortisol levels. Next slide. This is the same concept, but looking down to individual injuries. If you look at the panel on the right, that is the injury descriptions of what the codes mean, and then they're kind of graded into how they typically bin out into minor, moderate, or severe injury classifications. Again, on the left, or the y-axis, we have our constituent of interest. So for all these panels, the glucose, lactate, and cortisol, we're not finding any significant differences across injury categories. And this may be pointing to the fact that much of this physiological state that the animal's in when we see them is representative of the conditions preceding the release of the fish. So what was its state when it was released from the injury, from the hook, as opposed to a physiological response to that actual release itself. Next slide. So ongoing, we're continuing to look at the relationships of both the individual traits of the animal, so its physiological condition, its fitness, its fat levels, its size, all those things, as well as environmental traits, so time out of water, soak time, temperature differences, all those sorts of things, through the hook releases and the influence on both the injuries and the viabilities and how those can be predicted. So we're currently working through kind of some random forest work on that and other analysis of that work. And I'm currently conducting some of that work under a master's program through the Alaska Pacific University. Next slide. So Joseph, if you can just hit the forward button twice, there's two other items that show up on this one. There you go. This is moving into the recreational fishery, the guided fishery. And we're looking to actually get some real estimates in the field on discard mortality rates for those fisheries. So the first step in that, and this is funded under a National Fish and Wildlife Foundation grant, and we have partners on that through University of Alaska Fairbanks, APU, and the Alaska Charter Association. But last summer, we did a questionnaire of the skippers in the fleet, trying to get a better understanding of the typical gear being used on the fishery, and the way the most common fish handling practices, so we could design a similar study to the directed one that we just described. And so we did 51 interviews of skippers, roughly evenly split between Southeast and Central. You can see the numbers there. And the results of that showed that, the majority of the hooks being used in the fishery are circle hooks, although there's still a fairly significant proportion, 25%, that are using jigs, which are commonly using a J hook, but not always. Hook removals, 54% of them are reversing the hook back out, and 40% are twisting with the gaff. And there's the reversing the hook's often done with the pliers, and the twisting is done with the gaff. For fish handling upon release, we also looked at fish handling aboard the vessel, but focused on the release. Roughly 65% of the industry is using support of both the body and the tail, but there's also some that are using just handling the animal by its operculum and then letting it go over the side, and some actually are still using just holding it by the tail before releasing. So that whole effort was designed to help us design a field component for this work, and we are still working to drum up some additional funds for some additional satellite tags, because they are very efficient in providing us with kind of not instantaneous, but near term, so like 96 days out, survivability estimates on these fish in a very effective way, as shown by the previous study. And we're hoping on the next slide here, please, Joseph, to and hit the forward button one more time. We're hoping to have a field experiment in early next year, before June, to actually investigate the predominant gear types and what sort of DMRs we're seeing in this fishery. So that work continues. It's in planning stages right now, and like I said, we have some applications for additional funding to get sufficient tag numbers out there, with an application right now to the MPRB. And that's the end of my slides.
Thank you, Claude. Are there any questions from the SRB members? If not, we'll move on, and we'll move on to the genetics and genomics section, and which is the last one, and I'll let Andy Jasonowicz speak to that. So Andy, go ahead.
Yeah, all right, hi. Yeah, so I'm going to be talking about our plans for research in the genetics and genomics research area. And that really consists of two main research objectives. So first, determining the genetic structure of Pacific halibut in the northeastern Pacific Ocean. And then second is the analysis of genetic variability among juvenile Pacific halibut in the Bering Sea and Gulf of Alaska. Oh, next slide, please. And so I guess, yeah, before you get into it, I want to go over a little background of some, fairly recent studies that kind of provide our, the basis for our current understanding of population genetic structure in Pacific halibut. And so in 2010, Nielsen et al. Conducted a study where they compared fish between fish collected in the Gulf of Alaska, Bering Sea, and Aleutian Islands. They genotyped 228 individuals at 9 microsatellite loci. So essentially, they sampled the genome at 9 specific locations. And they found evidence that might suggest that fish in the Aleutian Islands are genetically different from the rest of the stock, or at least there's some reduction in gene flow between the Aleutian Islands and the rest of the stock. And I think I have an animation there, so maybe hit the next slide. Okay. And so I guess what's important to note about this study is that their sample collections were made during the summertime. And so those sample collections may not necessarily represent breeding populations of fish or spawning groups. All right, so in 2016, Drinan et al. Conducted a far more comprehensive study where they included additional sample collections. I believe they had additional samples from British Columbia. Some finer scale sampling was done in the Aleutian Islands, and then they also included some sample collections that were from Russia that were done in the mid-90s. And so, and they also focused primarily on using winter sample collections that were made during the winter time. So they also analyzed 2 different data sets for this. So the first data set consisted of 828 individuals and 9 microsatellite markers, so that they could, at least their markers would overlap with the Nielsen et al. Study and be directly comparable there. And then a second data set where they included additional microsatellite markers to improve power. And also a set of those markers, 32 of them, were actually derived from genes that are expressed in the genome. And so what they found is that they were unable to I guess confirm the results of Nielsen et al. However, in the analysis of their second data set, they found that Attu and Petrel Bank clustered away from the rest of the, or clustered were, I guess, were evidence that they might be genetically different. And so, yeah, there's another animation. So maybe hit the next. All right, yeah. So, however, with these sample collections, it's important to know that these were the only sample collections that were done in the summertime, with the exception of the Russian samples, which happened to group with the rest of the stock. So, is it that these samples are grouping together because they were collected in the summertime, or is there maybe something with the additional microsatellite markers that were linked to genes that are able to discriminate those populations from the rest of the group? So next slide, please. So yes, so as far as our work is concerned, really it's going to focus on revising our understanding of the genetic population structure of Pacific halibut in the northeastern Pacific Ocean, using more samples that were collected more recently, and using newer genomic techniques. And based on previous work that's been done, a large part of that is going to be focusing on the analysis of any structure that might be in regulatory area 4B. And so this map here, I've created this map to give you an idea of what sample collections we have available to us. And these are the sample collections that we have that were made during the winter time when the fish are on their spawning grounds. And so a lot of these were used in Drinan's study, the Drinan et al. Study, with the exception of sample collections made in 2007. And then we also have additional samples from the Portlock area that were collected in 2018 as part of the study to characterize the annual reproductive cycle of Pacific halibut. And then the special winter charter that was done this past winter was successful, and we now have sample collections from ADAC and the Western Aleutians from 2020. Next slide. All right, so as far as the methods are concerned, we plan to use low coverage whole genome resequencing, and this has been successfully applied to population genetic studies in other species, Atlantic cod and Atlantic silverside, for instance. And so taking this approach, we expect to identify on the order of millions of single nucleotide polymorphisms, or SNPs for short. And these are essentially, single base pair mutations in an individual's DNA. We plan to sequence approximately 50 individuals per sample collection for a total of 600. And then we're going to sequence, shoot for a 5x sequence coverage target. And so essentially what that means is we're going to resequence the genome of each individual fish 5 times over. And that'll give us the ability to call individual genotypes at all of those different SNPs that we identify. And so really, in order to do this, we're going to be aligning those sequence reads to the Pacific halibut genome that has been recently sequenced. Next slide, please. So as far as the population genetics analysis is concerned, we're going to calculate fairly standard diversity metrics such as allele frequencies and test for deviations from Hardy-Weinberg equilibrium, and then conduct analyses to look at the population structure. So estimate pairwise genetic distances between these populations. FST is one of those. And since we have sample collections from previous years, we can not only look at spatial structure, but also potentially how genetic structure changes over time as well. So we can use those genetic distances to also look for test for patterns of isolation by distance. And then using taking more individual-based approaches, we can do things like clustering. So admixture is one program that's designed to do that. It's based on population genetic models, and essentially it attempts to estimate the ancestry of individuals, and you can use this to determine the actual number of genetic clusters you have in your data set. So for instance, if your genetic groups don't necessarily correspond to the collections that you have, you can use this to disentangle that. And then we also plan to use other methods such as principal components analysis and then k-means clustering. This may not make the same assumptions about the data that these methods that rely on existing population genetic models do. And then also population assignment testing to identify potential migrants is another thing we plan to do. So, and then I think hit the next slide button. Oh, sorry. So I had an animation on that last slide. So also we'll have genome-wide coverage. So that'll enable us to genome-wide SNP coverage for these individuals, and that'll enable us to scan the genome for signatures of natural selection. So essentially, look for SNPs that show elevated levels of differentiation. And by focusing our analysis to those, we might be able to really improve our ability to resolve population structure and detect migrants and so forth. So that's that's where the genomics aspect really comes in. So like I mentioned before, the Pacific halibut genome sequencing effort is complete. It was completed earlier this year in collaboration with the French National Institute of Agricultural Research and the University of Washington. And I just have some statistics regarding the genome here. It consists of 24 chromosomes. It's approximately 586 million base pairs in size. And a large proportion of that assembly is actually in those chromosomes, which is a good indicator of quality. Our collaborators in France identified the locations of 23,625 genes, and which is going to be a great resource for us. If we do find regions of the genome that we're interested in interrogating further, we can use the positions of those genes to infer functional significance and what have you. The genome has been uploaded to NCBI's public data repositories, and so it's publicly available at the moment. And we have also requested an annotation from NCBI. So not only will we know the locations of the genes, but the function of those genes as well. And with this sort of resource available, it'll be a great resource to have to potentially develop SNP panels so that we can integrate genetics and genomics to addressing other research questions. And for instance, you could develop a SNP panel to geared towards things like population assignment, kinship analyses, and tailored to whatever research question you have. So next slide, please. And then finally, this I guess the second research vector we have is the analysis of genetic variability among juvenile Pacific halibut in the Bering Sea and Gulf of Alaska. And really, this is to infer the potential contribution of fish that might be spawned in other areas to the Gulf of Alaska and Bering Sea. And so this will tie in nicely with Lori and Ray's larval advection modeling work. To see if we can find, genetic signatures or that there might be evidence of fish from the Gulf of Alaska being, advected into the Bering Sea and utilizing the nursery areas there. And so to do this, we have fin clips collected from the NIMPS bottom trawl surveys in the Gulf of Alaska and Bering Sea. And we'll take a similar approach to the population genetic studies as far as the sequencing is concerned, but we'll be comparing genetic diversity metrics between these two areas, and then also estimating those admixture proportions for individuals between these areas to see if again we can detect those genomic signatures that might suggest an individual did not originate in the ocean basin that it was sampled in. And I believe that's it, unless we're going to talk about the So.
Yeah, thanks, Andy. So that concludes our presentation.
Just an observation, Andy?
Yep.
Yeah, thanks. Nice slides and very interesting work. I guess as you move forward, where you have an opportunity to incorporate samples other than the two primary hypotheses about five sites and then the juveniles, I guess I would argue that having the genomic resources and at least having some partial data in areas of growth, reproduction, maturation, and other things, it might not be a complete data set, but it gives you an idea of a whole genome sequence of individuals, say, that are growing at different rates, that you have transcriptome data for, et cetera, would be really, really valuable to have, where you'd have a small data set and it would be proof of concept moving forwards to incorporate the genomics data into each of the other areas within the biological research program's agenda.
Yeah, no, I mean, absolutely. Yeah, I think that would be great. And it would definitely be a great addition to those studies. And yeah, perhaps, I don't know if, we could develop some kind of a SNP panel to make, the sequencing of that not so quite extensive, but it would be, definitely great to do going forward. So.
Can I ask a few questions?
Yes, please.
Great. Thank you so much for the presentations here about the biological program. This has been really interesting for me and really helpful to get a sense of the incredible breadth of what you're doing here. I wanted to focus, just ask about a couple of things that that were not part of it that that maybe you've done before, and in that case, I can leave it there. But things that might go into the stock assessment model, for example, natural mortality. Has there previously been a focus on sources of adult natural mortality? Or are there important questions there?
I think probably Ian will step in and probably provide more meaningful information than I can. We have not been looking at natural mortality per se. We've been perhaps the only component that we've been investigating in our biological program has been mortality associated with discards in the different fisheries. But
right.
No, we have not, to my knowledge, and I don't think we've directly addressed that question before, but I'll let Ian, most likely, who's the person probably better suited to answer that question.
Yeah, I mean, maybe Ian could just tell us a little bit about whether natural mortality is an important uncertainty in the model, and if so, then maybe that might be a useful focus.
Yeah, thanks, Josep. And Olaf, yeah, natural mortality is definitely an important source of uncertainty in the stock assessment. It was something that we focused on quite a bit in the last round. And in fact, we considered adding some additional uncertainty due to natural mortality to the ensemble in the form of additional models. It's probably one of the leading sources of uncertainty that's not addressed in the model. The most recent real research push to investigate natural mortality was during the pit tagging study, which occurred in the early 2000s. And so we had tens of thousands of fish out for long periods of time. And that actually did give us some information on natural mortality, of course, conditioned on our understanding of tag reporting rates, tag shedding, all the other factors that go into tagging data. We have used that to develop a prior on natural mortality, and we've explored using that in various models, although it turns out to be relatively uninformative, and so we've either used a uniform prior or we've used fixed values of natural mortality. It kind of hasn't solved our challenge there, but I think it's certainly an avenue of that could be helpful. I think, like everybody else, we struggle to identify tractable research topics that would really give us something perhaps more than we got from the pit tagging study.
Yeah, right. I mean, once you're maybe, I don't know if this is quite right, but burned once on a big tagging study in terms of its ability to constrain natural mortality, it's you might have cold feet about jumping in again. That certainly happened over here in the Northeast in a few stocks. So was the issue that the estimates of natural mortality from the tagging study were so poorly constrained that it just gave you a very uninformative prior?
Yeah,
or was it that they were actually off from what the rest of the data was saying is plausible?
No, actually, they were fairly consistent, maybe slightly lower. From memory, the mode of the distribution was somewhere around 12%, and we use our current estimates range from, I think, 14 to 21%. So they were definitely in the ballpark. The pit tagging study had some other challenges, particularly with regard to some of the western areas and very low tag recovery rates. So it was certainly not precise enough to just give us an estimate of mortality, natural mortality, that we were comfortable fixing in these models. We had used it to condition some of our analyses and to generally provide us with a bracketing of uncertainty. But beyond a tagging study, it's hard to know exactly how to tackle natural mortality, particularly because I think at least for adult halibut, we're not even necessarily clear, and maybe Josep and his team have some better ideas than I do, but I don't think we really have a great idea of what the sources of natural mortality even are.
Well, what about some of the life history-based proxies for natural mortality? Have you guys looked into those? Are they useful?
I've done a little bit of looking at some of the various cross correlations between life history traits. One of the challenges we have for halibut is that much of that stuff is dependent on growth. And a lot of the rest of it's dependent on temperature. And of course, halibut inhabit a wide range of temperature from 2 to 10 degrees. And with regard to growth, halibut growth is unlike many other fish in that they grow at certainly different rates in different parts of the stock, but they grow nearly linearly throughout their life. So, and with a huge amount of variance about that growth. So our largest fish tend to be not even our oldest fish. They just are on a very high growth trajectory, but our oldest fish show evidence that they've been growing steadily throughout their entire life. So I've always been a little bit concerned that the sort of meta-analytic life history correlates are not terribly applicable to halibut because it's a bit of an outlier in some dimensions with regard to, say, von Bertalanffy growth.
Great. And that, thanks for that, Ian. That was really helpful. And that brings up, I guess, one of my other questions. I didn't see much focus on otolith analysis, and I was wondering, for example, the question that came up about looking at historical growth patterns, that strikes me as one that could be approached with analysis of otolith annual growth increments, or otolith microchemistry. Are these things that the biology group has looked at in the past, or is there a reason that they're discarded?
Yeah, no, the research group has definitely conducted those studies in the past, and in fact, hopefully in the next meeting in September, we'll be able to provide you with the results of a growth increment study that we're currently analyzing. From the information that we have right now, we can say that incremental otolith growth is definitely, for Pacific halibut at least, is not a good proxy for growth, for somatic growth.
Oh, too bad.
Yes, as opposed to some other species. So we'll have definite results on that. So there's a little bit of research relating to otoliths. That's been mostly done in the past, though, and we have done in the past some microchemistry studies relating otoliths, and those were done by Tim a few years ago. I think we have now our pretty much our bases covered at the moment. Our hands are tied in a number of projects. Those are tremendously interesting, and we would hope to at some point incorporate trace element or even stable isotope work to the work that we're conducting in migration. So linking, physical tagging with genetic tagging, and also with chemical and isotopic signatures in otoliths. That would allow us to provide. But if Tim wants to step in and very, very, very briefly, please, just comment on the work that you did most recently on otoliths, just to give a brief summary to OLAP on the kind of work that it's in fact in a publication hopefully soon.
Indeed, this is Tim. Tim has become Claude on your screen, and Claude has become Tim, but I'm still Tim. Basically, what Joseph first alluded to is an increment study that we're working on right now. The bottom line on that is that while individual increments for individuals do seem to be linked to individual growth rate, halibut that are half the size they used to be now have otoliths that are the same size as they used to be. So long term, the accretion rate doesn't seem to have changed as growth rate has changed on a population level. So that's where we can't really reconstruct historical growth patterns from otolith size.
Except within if I got it right, except within an individual lifetime, right? You said that within it.
You'll seem to see variance within annulus size that's likely related to their annual growth. It's just that those don't scale then to decadal scale changes.
Yeah, sure. But I would think that, I mean, we can talk about this offline, but I would think that you could probably use individual intercepts to deal with that in a mixed effects context where you look at growth within individuals, relative growth in warmer and colder years to get some understanding of environmental controls on growth.
Yeah, and you may very well get to that. And so to jump quickly to the next slide, we are looking into hopefully doing oxygen isotope analysis by
good.
annulus to reconstruct sort of thermal history and then link individual growth within years and over time to temperature. And then hopefully we'll be able to get nitrogen analysis on those annuli as well. That'll be dependent on sort of how much otolin is in the otolith matrix for it's species specific. So for some species, you can get enough nitrogen from an individual annulus to look at individual fish. Sometimes if they're not very nitrogen rich, you have to pool numerous fish within a year in a region in order to get sort of a diet signal. But that's definitely something we'd like to move toward, is then linking temperature and diet to individual growth rate and try to partition these two things out so that, for instance, as Ian's talking about and Sven has mentioned, diet constraints and temperature constraints, what sort of proportionally does that do in the real world linked to what we're seeing in a lab environment? Which of those is really driving growth? And it might change in time and it might change in space. And hopefully we'll be able to see that in our otolith records.
Great. I would love to talk with you more offline about the nitrogen part. I've been working with a geochemistry group at Princeton on a technique for recovering otolith nitrogen, and it's a bacterial-based technique, and it can use very, very small quantities. It might be effective here.
Yeah, and in the otolith conference, people were talking about that too, not just net nitrogen isotope concentration, but being able to isolate individual protein markers as well within otolith. So that would be a really interesting way of looking at things. Again, I think our baseline has to be to do some nitrogen analysis on the otolith to figure out how much nitrogen we got and what we can actually resolve.
Yeah, great. Sounds very interesting.
Yeah.
One other comment on otoliths, and it wasn't really clear if David was interested in the SRB commenting on the otolith section, which was a part of the report, SRB9, but wasn't presented today. But I mean, just an observation that you can have an oxygen-18 signature that would be associated with temperature by being, well, for several reasons, including being in a different place, right? Not just being at a different depth, and or because it's just a warmer, colder year, right, for growth. And I'm wondering if there is any other isotopic signature that you could simultaneously collect that would give an idea of geographic origin. So strontium, something else that may vary spatially across the area. So you could actually ask the question, well, is it just a warm year, or were these individuals at a particular time just in a different place?
Yeah, and that's also something that Joseph had alluded to with the paper that we're working on at the moment that should be out. We did trace metal and oxygen carbon isotope analysis on some H3 individuals looking for spatial signals. They're there. Unfortunately, it seems that the oxygen and carbon are the best spatial signals. We get a little bit of signal from manganese, and the trace metals don't really do much of anything. So trying to resolve sort of that difference between depth, regional temperature, and broad scale location will get a little confounded, because we'd probably be still just looking at oxygen and potentially carbon. So I'm not confident that otolith chemistry will give us an ability to resolve sort of fine scale spatial reason for the temperature differences. We did years ago also did a bomb radiocarbon analysis that demonstrated that some of the bomb radiocarbon signatures differ between the Gulf and Bering, so we might have basin scale ability to use C13, but that's probably also spatially pretty large scale.
Yeah, and it wasn't really explicitly mentioned, but based on what was described, I'm assuming that you're using a laser ablation where you're able to target multiple points, annuli, for instance, of the same individual, rather than total otolith ablation?
Yeah, our original work 10 years or so ago was whole otolith, because we essentially didn't have the techniques. We'd probably use micromilling, but if we can use laser ablation or microprobe, any of those three should be effective. And again, it's just, for some of these analyses, it's a matter of whether or not you can get enough material from an individual annulus, or whether or not you need to do more. All of those microsampling techniques are what we would be hoping to use.
Okay, thank you.
Okay, thank you. Thank you for the
Okay, hearing nothing, Tim and
presentation.
Carl will go back to being their former selves.
Yeah. So that, I think, brings us to the end of the presentations. We're an hour past the SRB drafting session. What I suggest, so our tasks now are, I think, for maybe the SRB to meet in camera for just enough time to go over the draft report so that we can get it back to the Secretariat for a review, and then we can all meet and go over it and approve it in the end. So we have potentially a couple of hours to do this. So I think what I propose to do maybe is have a 30-minute SRB drafting session. And then, well, yeah, I think a 30-minute SRB drafting session, and then come back at about 1:45 as a group and review it. How does that sound?
So just to clarify,
I didn't hear you. I heard just to clarify.
So just to clarify that you're going to spend the next hour together as the SRB and review and update the version which I sent back to you early this morning. And then at 1:45, your intention is for any interested parties to rejoin with the SRB to go through the report. Is that correct?
Somewhat. I think, I don't think we need a full hour for the SRB. I think a half an hour and then maybe some time that we can grab some lunch or something.
Sounds good. Okay, well, we'll all vacate the meeting.
So we'll send the report, the draft report, to you in about a half an hour that you can then circulate to your group.
Might be Ed or something. Sorry, say again?
We'll send the draft report to you in about a half an hour, say 1:30 at the latest.